Hi, everyone. With the Olympics going on right now, I thought I might discuss the positive effects of exercise on the body and on the mind. This week, I read an article that studied how long-term exercise can help reduce anxiety in a study of over 400,000 people. So, as we know, anxiety usually develops in adolescence, and it is estimated that up to 10% of the global population suffer from this mental illness. So that begs the question, in what ways can we prevent the onset of symptoms, and how does physical activity ease the burden of mental illness? So the researchers that did this paper, they used an observational study that followed over 400,000 individuals for up to 21 years. And then they also used R statistical software for their analysis. What they found was that a physically active lifestyle, in this case, they were skiers, is associated with around a 60% lower risk of developing anxiety disorders compared to matched individuals from the general population. And to conclude, the results of this study support the recommendations of engaging in physical activity to decrease the risk of anxiety in both men and women. Thank you.
Hi everyone. Today I have a short article that I would like to share with you. The name of this article is An Electrically Charged Thin Film Patch Used to Promote Wound Healing by Bob Yurka, January 28, 2022. Now, humans have been researching ways to promote healing for thousands of years. Throughout their research, they found out that electricity helps. However, due to the bulky machines that are needed to generate electricity, it is theoretically possible but hard to apply practically. Recently, the University of Electronic Science and Technology in China managed to develop a thin patch. This patch is only contains only 4 layers. It is only 0.2 millimeters thick, but most importantly, it is capable of applying electricity to the area that it is applied to. In their experiment, they found out that applying the patch allows mice to heal wounds significantly faster than not applying the patch. Even though there is no information about human trials yet, this piece of technology is still very promising and worth keeping an eye on.
We are going to be talking about advancing stem cell medicine. So this publication titled Transplantation of Intestinal Organoids into a Mouse Model of Colitis by Wantebe et al., published on February 2nd of 2022 in Nature Protocols. So essentially the crypts of the colon harbor intestinal epithelial cells that can be harvested and grown in gels in a way such that they self-organize into these sphere-like organoids with these protrusions. And so essentially they mimic our own intestinal tract because these regions here are the proliferative-like cells of the epithelial cells. And here we have the more differentiated cells like what we have in our villi that are like this in our colon. And essentially these researchers invented this transplantation using this organoid a few years ago, and their protocol has been replicated a lot. So they wanted to see how the different labs use their protocols, what were some similarities and differences, and are there any key techniques. So they noticed that many different labs around the world will do the similar process of using some sort of agent to damage the tissue cell. And then they add these organoid stem cell fragments that they take once they take from the crypt, grow the organoid, then they fragment and insert it into the part of the tissue that they damaged. And then those cells eventually, they bind to the base membrane, then and they bind to each other, and over time they start to kind of like start behaving unified and integrated into the tissue. And all the labs around the world do this, and often there is more than this process can be replicated more than 50% of the time. So this is overall a reproducible protocol. But what they do notice some differences is that different places around the world will do different parts of the intestine for this engraftment study for different purposes, like using regenerative medicine, studying development, or modeling cancer, but they wanted to focus on the regenerative medicine. And they started comparing different chemicals to use for the injury sites. So they will look at either DSS or EDTA, and they have different pros and cons, but they think that DSS is better for modeling colitis than EDTA because EDTA has specific purposes for transplantation that maybe possibly could bias the results. And what they noticed in the organoid is that the LRG5 is highly enriched in these crypt-like domains. But overall, these structures are generally heterogeneous and may be disrupting the success. And but having the expression of key marker genes is not necessary for transplantation, but they think that they want to have more homogeneous gene expression in the future by kind of creating an induction system. And also they noticed that younger mice are not sensitive to the DSS treatment for damaging, and older mice can have adverse effect. So they use mice for between 24 to 42 weeks old. And they use a dissection microscope to assess a 46.5 success rate that they think can be enhanced by improving the protocol or improving practice with the method. And they also can't do colonoscopy before and after, but they do histology. So here they see with this cadherin and the lysosome that the tissue is kind of integrated, binding to each other, and is just part of the tissue and functioning.
We'll be speaking on how the gut microbes are affected by the nervous system. So the article is called Study Shows How the Intestine's Nervous System Affect Gut Microbes, and is written by Karen Gulliman. So some background information is that for years, scientists have known that the gut bacteria are essential for digestive health. Other research has also found a substantial link between the stomach and the brain. So the research experiment was performed by Christy Hamilton, where she suspected that diseased zebrafish had issues with their gut pH. And she also gave acid-sensing dyes to zebrafish larvae. So the ones without gut nerves had more acidic guts. This resulted in an excess of dangerous Vibrio bacteria. So the conclusion for this article was that these findings imply that gut nerves regulate the pH, which helps in maintaining a healthy bacterial population in addition to controlling contractions that keep moving.